Then we'll have Nikhil Munshi from the Dana-Farber come up to the podium. He's been very much involved with all different kinds of research, but he was particularly involved, for example, with the first CAR T product that was successful, published in the New England Journal of Medicine and approved by the FDA. And so, as I said earlier, we want to have people speaking to you who have had hands-on experience with these things. So he knows all the details about the benefits, the side effects, and the like. And so please welcome Dr. Munshi.
Thank you. Thank you so much, Dr. Dury. And it's such a pleasure being here and also part of this wonderful seminar today, day and a half seminar to sort of give out the message about where we are. And the good news is, and I've done this for many, many years, I won't tell you the numbers, but quite a few decades, and it's so different today. And I'm almost hoping that maybe one of these days soon, we will not have a session on relapse myeloma, because nobody would relapse. So that's our hope, that we only talk about the newly diagnosed treatment. And what may lead to that is the newer advances in immunotherapy. So these are my disclosures. We work with everybody, so then you put everybody's name. And that's a good thing. There is no specific conflict. So immunotherapy, this is a complex slide, but what I wanted to convey by putting this out is that there are very many types of immunotherapies that we have been all working on, and literally for the last 30 years, from developing vaccines for myeloma to developing antibodies for myeloma. There are antibodies that directly kill myeloma cells, and there are antibodies that excite the immune system, etc. And each one of them has their place, and they are having some success. We are having some success. The one which is most effective and what I'm going to focus most on are the one on the right side, what says passive immunotherapy, where we are using our ability to change the immune cell and make it work better. So we take your immune cell or modify your immune cells and let them kill myeloma cells. And that passive immunotherapy has really revolutionized what is in myeloma in the last 3 to 4 years. So if we go through each of this treatment, there are drugs that can achieve immune responses. And 3 of the drugs you might be familiar with, thalidomide, lenalidomide, and pomalidomide, which are commercially available for many years now. There are 2 more which are in the pipeline. The last one has a number, but it also has a name called mezigdomide. And for some reason that I can't explain, maybe Brian can, the names are always very complicated. How they are developed, we really don't know, but the companies come up with names. So the mezigdomide is another one. They are the next generation of immunomodulators, going to be a very important part of treatment as we do it in the future. Then there are antibodies. There are 2 antibodies that Dr. Voorhees discussed for newly diagnosed patients, daratumumab and isatuximab. And then elotuzumab is used more at the relapse setting, so we have 3 antibodies. And then there are checkpoint inhibitors which are good for immune treatment. We investigated, it did not help in the old days. Now in the new days with all the immunotherapy we do, they are coming back again. How can we make better immune system by using them? And so there is no name there, but now just wait for a few more names to show up there. There is this belantamab antibody conjugate against BCMA. It was approved. For various reasons, it is again withdrawn. We are hopeful it will come back because it really works in a certain group of myeloma patients. What I'm going to talk about more focused is not those good things that are in the black here, but the 2 red lines, the CAR T cell therapy and the bispecific antibody therapy. And these are commercially available. This is what we now use quite routinely. So first, let's talk about CAR T cell. What is CAR T cell? How it is made? And how can we administer? So simple concept here is that we take the immune cell from patient, their own immune cells, and we genetically modify it. So what we do is we take the immune cell, called the T cells at the top, and then we use certain mechanism a viral vector, goes into the immune cell and gives the immune cell this new ability. There are new offshoots coming up, which makes the immune cell identify myeloma cell very specifically and kill it. That's as simple as that. In the process, the immune cell begins to divide. So it does two things. It kills myeloma, but also at the same time grows further. So that one cell becomes 2, 4, 8, and such. And because it needs to fight more myeloma cells. There are millions of myeloma cells in the body when the patient has a relapse disease. So both of these features makes it very effective. It can kill and it can grow to kill more. And so this is the simple concept of CAR T cell. And it's very straightforward, it is very standardized. And so when somebody is going to go on CAR T cell therapy, first step is to collect the cells from the patient. It is simpler than collecting stem cell for transplant. Stem cell for transplant is more involved, we give growth shots and everything. Here all we do is put IV line and collect some blood. It's, you could say, similar to collecting blood for donation. The second step is that these cells are then they go into the lab, either our lab or now in the company lab, and they are modified by the method that I showed just before, using the virus. And it takes around 4 to 6 weeks to prepare. There are newer methods, but it might just take short time, 2 days. But the current FDA-approved process is around 4 to 6 weeks to make the cells. Once these gene-modified cells, your own cells, are produced, they are shipped back to our lab and our clinic. And then we give some chemotherapy, small dose, and then give the cells back. That's the whole process. And when you get the cells back, they begin to do what they're supposed to do, kill cancer cell and grow further. So how do we decide what kind of gene to put in the CAR T cells? And it's based on our ability to find what is unique to cancer cell, what is unique to myeloma cells. Because we want the immune cells to identify only the cancer cell, nothing else in the body. Otherwise, we will have side effects. So here there are 3 names, LAMF7, CD38, and BCMA. These are all identification on the myeloma cells, unique to myeloma cell, not other cells. And the one which we have used the most and has the greatest success is the BCMA. You can only see those red marks, which are the myeloma cells. Everywhere else, it is low. So we don't get much side effect in the body. We don't get anything else except killing myeloma cell. And so that has become our target because it is so unique to cancer cells and not other normal cells. There are some side effects, but not major because of that. So there are 2 products available in the market right now. And we were all part of development of both in the clinic. So the first one to become available, it's called ide-cel, or you may know it better by something called Abecma. And this was a study that led to its approval. It was a 128 patient study. What I want you to look at is on the right side, up here, this is where we collect the cells. This takes 4 to 6 weeks to produce the cell. During this time, quite often, these are the patients who have more advanced disease when they went on the study. They had 4, 5, and 6 lines of treatment. So during those 4 to 6 weeks, patients' myeloma may not remain stable, so we may give some things to hold it, what we call bridging therapy. But when the cells are ready, we give this chemotherapy in low dose, give CAR T cells, and follow the patients. And just to highlight how advanced the patients were on this study, they had 6 various lines of treatment, not 1, not 2, not 3, 6 lines of treatment. And it ranged from patients with 3 to 16 line of treatment. So very advanced disease in this patient population. And the reason it's good to know that is because what was the result? And the result was that 80% patient as a 6th line responded at the dose that we would like to give to these patients. They respond very quickly. In 1 month, myeloma has come down. In 3 months, it goes to 0. In whichever patient it is going to go to 0. And it goes to 0 in around 40% of the patients. So very effective, very quick response. Now, the second part is how long does the response last? Now, these are the group of patients where, based on our experience, their ability to get response to anything else available was going to be around 15 to 20% at the most. We get 80% response with this. So really, totally unexpected. Number two, even if they respond, the response will last for a month or two. Here, at the dose patients get, the response lasts for 12 months and more. And so not only it works for a long period of time. And that's why we have so much excitement about this treatment, that it works very well and it lasts very long. And if you look at how long patients live, these are the patients who otherwise would be considered to have limited lifespan. Here, more than half of the patients are still doing well beyond 2 years. And this is still new study. It's still continuing because maturing. These are early results. So results are only getting better than the number I'm showing here. So that is the excitement where we would have not expected anything even for 2 months. Now 2 years and they are doing well. So that's one CAR T cell, a BECMA. This is second CAR T cell called ciltacabtagene autoleucel. The simpler name is CARVYKTI. That's what you would be more familiar with. So CARVYKTI is a little bit different type of molecule. I won't go into the details unless you have a question. But it also works superbly well. So a similar study as the first one I showed was done with this molecule as well. This was the second one. And here, the response rate is 98%. Literally everyone responded except one patient. And 80% of the patient, 82.5% of the patient, myeloma went down to 0. Again, these were the patients who had 6 lines of previous treatment and they had such a superb response. And if you look at this MRD negativity, which you have heard, I'm sure heard a little bit during the yesterday and today's session, half of the patients, their myeloma in the bone marrow is non-existent. And so very superb response. And how long this response lasts? It lasts very long. So you could see that patients who are MRD negative, the top curve here, they are out 2.5 years, and in them myeloma hasn't come back yet in half of the patient. And this line here are the patients who are surviving and surviving well for a long period of time. So incredibly deep response, almost everyone responds, and the response lasts for quite a long time in all these patients. So this is a reason why you could hear the excitement in Dr. Dury's voice about immunotherapy. And it is because of this result, even at an advanced stage we see. Now, this is a complex thing. I'm going to simplify in one sentence. Which patients benefit from this treatment? The bottom line is everybody benefits. Whether the patient has extramedullary disease, those risk factors you saw, high risk, low risk, almost everyone responds very similarly. So this treatment doesn't care it's a good or bad myeloma. It works almost everywhere. And common question from the transplant world comes up, am I too old to get this? And the good news is that's not true anymore. So here in this particular study, we looked at patients who were more than 65, more than 70. They still were doing well. The response rate was still 85 to 90% in this patient population. The toxicities were not excessive. I had 2 patients last month, 179, 180-year-old, went through it smoothly, very well, and myeloma within a month has gone to 0. So up to age of 80, if the health is good, we are comfortable considering this type of treatment. So the age bar has gone up much further. We can do this safely. Again, we look at certain features, but in general, it can be safely done in older patient population. And because of this great success, we are now beginning to do it earlier on. We don't need to wait till the 6th line. So in our clinical study, we are beginning to do early. So these are the patients who had only 1 to 3 lines treatment. Again, the response rate is same, 95 to 100%. And we are looking at patients high risk. So how does high risk patient do with this treatment? In this smaller study, the response rate was 100%. So now the high risk is not so high risk once we do this treatment. Of course, we have to see when does myeloma come back in this patient, can we delay it even more? But at least we have a very good beginning in the early stages of this study. So this is the very broad applicability. Now, everything has a price. The price here is small. Now, the price I'm talking about is side effects, not the price on the financial part. That's another question for discussion. But as far as the side effects are concerned, And I'll have a slide to compare it with transplant. But there are toxicities, and there are 3 toxicities that we would keep in mind. I would discuss quite in detail with my patients. One is this called cytokine release syndrome, and I'll discuss in a minute. Then there's a small proportion of patients who get what is called neurotoxicity or neurological issues. And then there are infections-related problems, and that actually is something we have to be very aggressive about because it can affect your immune system. And we need to give prophylactic antibiotics, we give IVIG to prevent infection. So what is the first one? Cytokine release syndrome. So what happens is that when we give the immune cells, the CAR T cells, as I suggested, the CAR T cell grows. And they not only grow, they grow 1,000-fold, 10,000, sometimes 100,000-fold. We need it to kill all myeloma. But because they grow so fast, they produce substances in the body as a natural process, normal process, and that sort of overwhelms our system, the normal product produced by the immune cell. And because of that, we see various features. Now it's very small written, it might be in your printout. But basically patients will get fever. That's the first features of it. And when patients develop this kind of reaction, and 90% of the patients do, it tells them that their CAR T cells are working, they are growing, and they are killing. So quite often, patients are very happy when they get fever because that means things are happening. Partly true, partly not true. Not true in the sense that there are patients who do not get fever and still respond well. If they get fever, they're going to respond, but if they don't get, so if somebody doesn't get a fever, I don't want them to be disappointed. It's still working, except you're lucky not to get the overwhelming issues. So fever is one. We keep an eye on blood pressure. We can keep an eye on cardiac function and oxygenation. Majority of the patients, as I will show the data, they get it, but they get very low level. And we have drugs to treat it. And what has happened, what is important is that the centers that we all work in, we are very used to handling this. And that's an important concept, that be able to manage this safely. We have drugs that we can give to knock down the chemicals that the normal cells produce, and so it doesn't expand into any blood pressure changes, etc. Steroids work as well, and there are various other manipulations we can do. So we can now identify it early. We know we can treat early without affecting the outcome. The cells will still work even if we treat it. But that's a side effect that we watch for very closely. That's one of the reasons why you will be told to stay close to the hospital for a month or so, because if there is any issue, you can come immediately, we can take care of it, and it becomes safer. The second side effect is this neurotoxicity. I wouldn't go into too much detail because it's not frequent. It happens 10 to 15% of the time, and it is usually very mild. Headache, lethargy, etc. And when we are in the hospital for 2 weeks, you have headache being in a bed doing nothing. But when they complain of headache, we take it seriously. It's not trivialized that, oh, it's just a headache, take Tylenol. We pay special attention because we need to nip it in the bud. And doing that, it becomes safer. Similar treatment for lymphoma, there is a greater problem with neurotoxicity. In myeloma, we are lucky we don't have as much. But then there are questions that are asked in the bottom, and there are very straightforward questions, and patients know it so well over a period of their being in the hospital that when the doctor goes into the hospital, patient answers all the questions without being asked about 3 names, how to go 7 minus 100 or successive numbers that come up. But these are the simple tests which tells us, is there any signs of neurotoxicity which we need to treat? And then we look at the real data for the 2 agents that are approved. 95% with one drug, 84% with second drug get the cytokine release syndrome. So almost everybody does, except only 5% patient, it is grade 3, where it is serious. We quite often transfer patient to ICU to manage certain blood pressure, oxygenation, etc., sometimes just as a precaution, but very small number. It happens quickly. With one drug, it happens in 1 day. With the second one, it takes 7 days. And that's important because we know when to expect it. The neurotoxicity, 17 and 18%, but only 2 or 3% are grade 3, and so they are minimal. The one which we have to be very careful, and I'm stressing it because it's easy to fix, it's easy to prevent infection, but it's if we suspect it. So we give prophylactic treatments, and your doctors who treat would be very familiar with it to how to prevent it. So the blood counts might stay lower, and as Dr. Voorhees suggested, sometimes we may end up using stem cells to bring your blood counts if they remain low for too long. Doesn't happen very often. It's less than 2% or so where we may need it, but we may have an ability to overcome it. And rare patients get what is called delayed neurotoxicity, happens after a month or so, and we have begun to know how to prevent the late neurotoxicity. Now, the 2 CAR T cells are approved. We have a list of various other, and I'm not going to go through each of them. They all have similar promise, works close to 100% of the time, works very quickly, works deep. Each one has some benefits and some newer nuances to make it work longer and better. So there's tremendous hope of what is coming about in next few years. So what the overview of CAR T cell administration? We identify a patient who is eligible by various criteria. We evaluate them, make sure they are good selection. Take this apheresis means we take their blood collection and send for the lab processing. And once the lab processing started, we may give bridging therapy, then give this low-dose lymphodepletion, and eventually they get infusion and they're monitored. Straightforward process. So just to give a quick update on which patients are to be considered for CAR T cells, the first and the only important criteria today is that patients should have had 4 lines of treatment, because that's what the approval is for. So sometimes you might end up getting most of the good drugs in the beginning in the first 2 lines. We still may have to do something alternate for 3rd and 4th line before we can go to this treatment. That's how our system works. Believe me, it is better than the rest of the world. My colleagues who we work with, all of them in Europe and other modern Western countries, they don't have access we have. So even though we all quibble about it from physician side, and we are all with the patients that I want to give it sooner, I think we are still better off. The good news is, as Dr. Voorhees said, there are very good results coming about from some of the studies which show it works very well, not surprisingly earlier on. So maybe in next few months to less than a year, we may have approval to do it sooner, not have to wait till 5th line treatment. But currently, that's what it is. And patients should have received an immunomodulated drug, a proteasome inhibitor, and an antibody, which almost everybody gets. Even with 1 line, now they get all of this. The next thing is, usually we want the disease to be progressing. And I think that is clear. We don't want to change treatment if it is working. But if when it stops working, we change it. And finally, as I said, age is not the criteria. We look at the physical condition rather than age. And even up to 80, we have very safely done it, and even a little bit beyond 80 in select cases. Unlike transplant, having perfect cardiac function is not as much of a requirement. We do test it. We want certain level, but not same as transplant. Kidney functions, and the studies was different, but now in real world, we do CAR T in patients with kidney dysfunctions. We had 2 patients treated on dialysis with CAR T successfully and safely. So we can do it with right precaution, right experience, etc. Infections we have to prevent. If patient has an active infection, that would be something we have to very carefully consider. And the patient's immune system is important, because after all, we are using your immune system to treat. If you don't have good immune system, we need to modify it. So because of that, patients should not be on a chronic steroids or chronic immunosuppressive agents, and should not be getting too much blood thinner. Certain level is fine, but we have to know and modify it. And finally, having normal blood counts for commercially available CAR T is not a must. We can utilize in patients even with lower blood counts. So those are the benefits now of having commercial CAR T. And if we differentiate CAR T versus transplant in very brief, I would just say that age, unlike transplant, age is not an issue. And unlike transplant, we do not need a high dose chemotherapy. What we give is very low dose. So it's much less toxic. Most of our patients have a transplant, and I've always asked them question, what do you think your transplant experience versus this? And almost uniformly, everybody says, oh, this was much simpler. It still sounds daunting. You have to be in the hospital, etc., but it's much simpler and much safer. So this is about CAR T. Now, the second equally exciting part is the second immunotherapy, which is called bispecific antibody. So you heard about antibody treatment like daratumumab and isatuximab, etc. So how are those antibodies different than what I'm going to talk about? And there's one big difference. So what these antibodies do is that this yellow antibody is against BCMA, which identifies myeloma cells. This is how myeloma cells is recognized. This blue antibody was developed a while ago that identifies the immune cells. So in the modern, the advances that happen in technology, we are now able to make an antibody which combines the two blue and the yellow ones. And the combined antibody, here does two things. This part of the antibody identifies myeloma. This part identifies the immune cell. And in the process, it brings the two of them together. So it brings the enemies together. And when the immune cell goes so close to myeloma cell, by its own characteristic, it kills it. So by using this antibody, we're able to bring the immune cells close to your myeloma cells and be able to kill it. And that's as simple as that. The process of making it, the technology is very complex. For practical purpose, it is as simple as that. So we have a commercially available now, FDA-approved antibody called teclistamab. It's off the shelf. Unlike the CAR-T, I don't have to make it. It's in the bottle. I can just take it and give it. So this is off-the-shelf product and does the same thing. It binds to myeloma cells, it binds to immune cells, and leads to myeloma cell killing. And based on very good early safety and other results, this study was performed where it showed that in, again, advanced patients had 5 lines of treatment. 63% patients responded. Proportion of patients, half of them had complete response. So very advanced disease with a simple antibody, we get very exciting results. And if you see further, responses don't last for 2 months, they last for a long period of time. Here, 12 months of before the disease progressed in half of the patient, and this number is still increasing. It was 1.5 years of overall survival. So again, it works well, works for a long period of time. And if you look at the toxicity, they're more or less similar to CAR-T, but a little bit lesser degrees. Again, it is because we have learned how to use it safely. We start with a low dose, and over a period of a week to 10 days, we increase the dose. In our experience and everybody's experience, it is safe. It makes it safer. The cytokine release syndrome, although it happens in majority of the patients, 75% of the patient, it's of the lower grade. We can manage well. The other side effects, I won't go through it, but they're also mild and tolerable. The only, again, caveat is to keep an eye on preventing infection. So this is an off-the-shelf treatment, works tremendously well, and it's one of those immune-mediated treatments. There are 2 antibodies which are expected to be, we are hoping they will be approved for myeloma in 2023. One of them is this one, elrantimab. It also again targets BCMA, similar to teclistamab. In the studies, it shows 65% response rate. This antibody is given every 2 weeks. It's a subcutaneous injection, so it's relatively easier, less frequent. Works very well. And so, again, great excitement about using this antibody. And importantly, the side effect profile for it is also very similar. 75-80% patients get cytokine release syndrome, but of a lower grade. And so we can manage it very well. And by now, most of us who do this for a living, we know how to manage this and we can make it safe. So this is the bispecific antibody that is likely to be approved, we are hoping in this year. There's another bispecific against a very different target. The target's name, like the BCMA, the name is GPRC5D. Its commercial name, or not commercial, but technical name is called talquetamab. And this one works differently because the target is different, but the principle is same. It brings the immune cell close to myeloma cell using a different recognition signal. And again, it works in 70% of the patient with very similar quick effect. Overall responses are quite high, and patients respond very quickly within a month. So again, we're going to hopefully have this one also available commercially within 2023. And the interesting part is that even if the other treatments stop working against BCMA, we may have a totally new option to go to after the another target. So we have one more new treatment coming up entirely different than whatever we have been doing for the last couple of years. So very, very exciting new development. And the third one, I mean, the list goes on, and that's the good part for all of us, is this another target called FcRH5. The molecule is called cevostamab. It has also efficacy, 60% or so, slightly lower, but it still works well. It is a bispecific antibody, so it's same immune mechanism, similar side effect profile, but very promising efficacy. So we may have 3 different targets against all this molecule, and I think we will stay tuned for one of these 3 or 3 of these 3 being approved in the current year. This is a laundry list, and it's deliberately put into smaller letters because I don't want you to necessarily read it, although you're welcome to. It's a summary of bispecific data that was presented at ASH meeting 2 months ago. It tells you that I showed you 3 or 4 of them, but there are many more. They all, the red box, the first red box here, says that it all works 60 to 70% of the time, whichever the target. So very effective. And then we see some of the toxicity profile here. This is highlighted is that we need to keep an eye on infection-related issues. Some of them have less issues, so those, when they're approved, they may become important earlier component of our treatment. And so, again, this is the reason for excitement. And finally, till we cure, we can't stop. And so we are still continuing to improve upon what we have today. And these are various things which are being done. We are trying to produce CAR T cell with newer methods. We are producing CAR T cells with newer abilities so that they can kill more, cause less side effects. We are using CAR T cells that can persist in the body longer time. So they work on their own as a maintenance for long periods, so myeloma doesn't come back up. And then the various other approaches that we are. The most critical approach that's not listed here is that we're also beginning to learn how to combine CAR T with the current treatment. Can we combine it with daratumumab? Can we combine a CAR T with bispecific? So we have a lot of very important, interesting studies ongoing or to be started to put all this in the perspective. And the bottom line message I would give as a conclusion is that these immune-based therapies are incredibly, they provide really unprecedented level of response. I've been doing this for 30 plus years and have not seen something like this. So effective at a late stage of the disease. So I think what we have to build on is now, the only little task left is we can get rid of myeloma using the technologies we have and coming up. If we can prevent it from coming back, then that's a cure. And I think we're almost there. So thank you very much.
So thank you very much, Nikhil, for this excellent presentation on the exciting immune therapies. So we're going to have a panel discussion where we'll actually all sit down again. And so I'll invite our other participants to come on up. Yes. In Iceland, you have all these therapies as well. Let's see. Yeah, we have enough chairs. We can all sit down.
You can come
Yeah,
here,
okay.
not come here.
All right, so we have this final kind of open discussion about the exciting immune therapies, but we can also comment on some other new trials or drugs that you maybe have heard about, and we'll try our best to tell you what we know about them. Thanks. Okay. Do we have questions for Nikhil, for example? Very good presentation, apparently. Yes. Okay, there's looks like one coming back there.
Yeah.
Right here.
Okay. Steve Weinstein, okay.
Thank you, Dr. Dury.
He's a leader of a group here in Florida. Just to be
What
aware.
about me? One of the things we hear a lot about is T-cell exhaustion. So I was curious if that happens, is there a way to recover from that? I was also curious about stacking different CAR Ts in case one fails. And then also, I believe, Dr. Dury, you were referring yesterday I think it was the GCO12F, the dual CAR.
Yeah.
So I would repeat the question. The question is, how do you prevent T cell exhaustion? So to explain what is T cell exhaustion is that the immune cells are supposed to do their job and kill tumor cells and everything. But like anything else, like we, after doing too much, get tired. And so the T cells can also get tired, and they don't do their job as well as they are expected to. And that is, in some ways, is called immune exhaustion. In reality, the cells change. They get some characteristics so that they can now rest without doing their job. And so it's a sort of a resting state for the T cells. And there are very many things being done. One of the research effort is to first select the cells which have less likelihood of going into exhaustion. We can take more T memory cells, T central memory type of phenotype, which have a little less exhaustion phenotype. Number two is to give some break, so that if you're giving bispecific, I think there is evolving concept that there is a time when we need to stop it for a little bit and then restart it. And the third thing would be that in CAR T world, which is still new concept, where whether we can reinfuse something so that the new non-exhausted cell can be given. And the fourth one there are a lot of drugs, including pomalidomide and the newer immunomodulator, which can overcome some of the exhaustion phenotype. Even the checkpoint inhibitors, which are actually the markers of exhaustion sometimes, the antibody against them can be helpful. So there are a lot of interesting lab and early clinical work ongoing. Each of one of them has a good possibility of overcoming exhaustion.
Right, just some comments. So in addition to monitoring for MRD, we're setting up panels to monitor for the immune status. And so we do have markers to identify if we have these exhausted T cells. we can see if those are building up. And so basically you have two options, some rest, and maybe try to whip those exhausted cells into action, or maybe to give T cells that are tuned up, and we talked about that before, maybe trying to harvest T cells earlier in the disease where you can have as active T cells as possible. But I think we're going to see more immune monitoring where we're trying to assess the pattern of exhaustion, but also we're looking at what is the pattern of patients who are doing well to see if there's a pattern where the immune system does seem to be doing its job.
I think they should go to vacation to Florida and
yes,
get better.
Yeah, we have some data on that, I think. Okay.
Hi, this is Paul from West Palm Beach, and my wife has a question. She's a little bit shy about it, but it touches upon a topic that was brought up a little bit earlier in another discussion, but I want to present it again because I think it's relevant. In regards to collection of the T cells,
right.
That's very new technology, and I know it's not approved yet. I asked the doctor, is Dana-Farber working on that? And if you could, look into your crystal ball and maybe give us an idea of when T cell collection may be ready for prime time. Thank
Okay.
you.
Yes, so the question is, when could the T cells be collected? And currently we collect T cells right before we are ready to make them into CAR T cells. And the problem can end up being that if patients have many treatments, then their immune cells are also a little bit affected. They're not as good. And theoretically, it can affect the outcome. And so the point being raised here is that can T cells be collected earlier, and would they provide a better outcome, better results? And so there are a lot of nuances to it. The answer is yes, they can be collected earlier. We are also working on, can we use the stem cells we have collected, which is usually very earlier on, and produce it. The companies won't. Our research groups like ours can do it, so we can safely produce CAR T cell using those. So there are efforts ongoing to think about it. The good news is that most likely very soon, within a year or less, in my hope and guess, the CAR T will become available much earlier as a first or the second line treatment, and then pre-collecting it may not be as necessary. So today, yes, I would like to. Hopefully within a year, we may not need to.
Yeah, so a comment on this is that obviously when harvesting is done and collection for T cells, we want it to be reimbursed. And the payers, the insurance companies, they do pay attention to guidelines. And so I'm thinking that, maybe we can talk about this, maybe we do need to have IMWG guideline to indicate that this is a smart thing to do.
Right.
And then that will have influence on the payers to allow us to actually do this. Okay.
There's a follow-up. Yeah.
All right.
Just a quick follow-on. You mentioned that possibly stem cells that have been collected could be utilized for T cell also. Is that?
I think I put my foot in.
I'm sorry. The reason I ask
You need a bigger biobank, a bigger boat.
So theoretically, when we collect stem cells, the stem cells contain also T cells or all different types of cells. So theoretically, what is in the stem cell can be repurposed to transduce the T cells from that. It's a theoretical possibility. In practically putting it into the patient back, it requires complex management of those stem cells. Commercially available products do not follow that algorithm for obvious reason. And so theory is good. Practically, that's not yet doable today. In the lab, yes, we can. In the patients, we can't.
Right. Okay.
Hi, I apologize if you've touched on this before. How do you decide between the Carvykti or the Abecma? How is that decided?
I can let somebody else answer. No, I can answer.
So from my perspective,
It's one thing.
whichever product is available to you now is the best product, to be honest with you. There are differences in the two studies with regards to the baseline patient characteristics, so I think it's a bit dangerous to compare the results of, the KARMA trial and the CARTITUDE-1 trial. Just for example, 40% of the patients in the KARMA trial had extramedullary multiple myeloma outside of the bones, which we've already talked about earlier is a high-risk feature. There is a much smaller percentage of patients with extramedullary multiple myeloma in the CARTITUDE-1 trial. So you look at 75% response rate versus 98% response rate, and you're like, oh my gosh, the decision's easy, I'm going Carvykti. It's much more complicated than that. I would say anecdotally, in the real world experience, since these things have been first FDA approved, my experience with the two products is very similar.
Right. So I think in the short term, whichever one you can get, take it. Sign up right away. Don't lose that slot.
Yes.
Right.
I think that moving forward, I think that this is a field that will rapidly evolve. And I think that, 5 years, 10 years from now, we will have new products. I think that manufacturing is an issue right now. We have manufacturing, which takes several weeks, and you need this bridging therapy. I think it's going to be fantastic if we can have this shorter manufacturing time. 2 days, 48 hours will be fantastic. I do have a sense that the T-cell products that have this dual head, you hear Silat mentioned where it was attaching to the myeloma with two arms, right, rather than one. And I think that we're going to see more of that, where a double approach to the myeloma maybe is going to be more helpful. So I think that next generation is going to be true in T-cells. You can see for IMiDs, we have thalidomide, we had Revlimid, we have pomalidomide, and now we have the CELMoDs. I think it's going to be the same for the CAR T cells.
Right.
Yeah.
Can I just add on a point? So in my center, we had a list of over 90 patients on the CAR T cell list. Now we have wider availability. Some of those patients are getting the teclistamab, which you heard was an off-the-shelf bispecific. We tend to think about people that are a little bit older or more frail, that are 75 or older, they might be better with Abecma because I think that the CRS might be a little bit more with the Carvykti. And so again, having that discussion with your team, getting on the list is a first step. And don't forget clinical trials with CAR-T or bispecifics if you're in that boat.
So maybe I can ask Sigurdur, what is the real world for CAR-T in Iceland, you know?
Yes.
I mean, because that's true for places all around the world.
Yeah, right. I think so we don't do it in Iceland at all. So we send our patients actually to Sweden for CAR-T.
For CAR-T.
Yeah.
Great.
Yeah, I mean, this is the real world where this access issue is really important. And resources need to come into play. You need to set up a whole center, mostly linked in with the transplant centers.
Right.
But I think, did you send patients for transplant to Sweden as well?
So allogeneic transplant we send to Sweden.
Yep.
Auto transplant we do in Iceland. So
Okay.
teclistamab and talquetamab we can do in Iceland, but no CAR-T.
Right.
Yeah.
Not yet.
Okay.
Not yet.
Not yet.
I think this gentleman had a question.
I have a question regarding, over here,
Okay.
regarding stem cell harvesting. If you did not get extra stem cells harvested prior to your transplant,
Right.
does it make sense to do that sooner rather than later, or does it not make sense at all to do? And then my second question is, to what
Right.
extent can bispecifics and CAR T-cell follow one another, and are they effective doing them multiple times?
Right.
Do we know that yet?
Yeah, I think, Peter, you touched on that. Yeah, I mean.
I'll take the second question
yeah.
first, and then maybe
Yeah.
you can repeat the first question. So can we give CAR-T and bispecific subsequently, one after other? And the answer is yes. So the CAR-T stops working in patients where it does stop working. A lot of patients are still not relapsing, but those who do, it's not because the BCMA is gone on the surface. Occasionally that's the case, but most of the time not. So they stop working for various other immune reasons. And because of that, teclistamab can still be very easily, safely given, and in quite substantial proportion of patients, around 50%, it can still be effective, similar to other populations. So yes, CAR-T and bispecifics can be given in a successive manner and can be given safely and in reasonable number successfully. And your first question?
Right. And as I mentioned, we are trying to study that with this immunotherapy database led by Dr. Tom Martin at UCSF, where we're trying to get real data, real world data, these magic words of what really happens when you do these different sequencing and the like.
So my first question had to do with stem cell harvesting.
Oh, yeah.
Get it done. Is there an option?
Right.
I'm sorry, I have to interrupt at this point because we are way over on time.
Oh, okay.
But the good news is, because I know that this is like, magic that we're being
All
given
right.
here. But we're going into breakout sessions now. So if your questions did not get answered, you
All
can
right.
select
What's the
the
breakout
breakout session that's right for you.
All right.
And
Well,
We'll do whatever we're told.
Wonderful. Thank you so much.
all right. So thank you, guys. Our class is dismissed.